All right, guys, we're back online to carry on with next three speakers. So right now I'm going to introduce Victor Greiff. Victor is currently a professor at Oslo University. He was trained at Humboldt and at ETH. And he's a very prolific group leader studying machine learning approaches to antibodies. Yeah? So this is like, he speaks at pretty much every conference that touches on antibodies that I have seen. Like, obviously, like, this one could not do without them. Yeah. And hopefully we're going to hear today about some of his approaches to study immune repertoires, especially the computational approaches for developability profiling. So, Victor, floor is yours.
Thanks, Conrad, for the nice introduction, and thanks, Conrad, Andrew, and Jen for organizing this great symposium. And thanks also to the numerous members in the audience, and especially to the ones in Europe that, because I know it's getting late, so thanks for sticking around. So I will be talking about developability profiling of natural antibody repertoires. This is a quite new topic to us, so most of the results that I'm showing, or all of the results actually that I'm showing, are currently unpublished. We're trying to put this on bioRxiv this year, so stay tuned for that. And some of the results are still preliminary, so although most of the things I'm showing should be quite final. I would just like to introduce the main contributors to the study in the very beginning. Dr. Habib Bashour, Eva Smorodina, Jan Song, and Matteo Parise. All of them have contributed equally to this study. So it's really been amazing teamwork over the last 2 or 3 years. So I'm going to talk about antibody developability. Mostly my lab is interested in antibody binding, but here we try to really focus on developability because it's, as we heard before in the previous talks, it's a huge issue for getting actually antibodies to the market. And we can define developability as making actually a drug out of it, as Pietro has also mentioned. Since binding is great, but binding alone is not enough. There are numerous biophysical and physicochemical properties that need to be taken into account, which you see on the right side of the screen, that make an antibody a good antibody. And so far, it's not really known what the best combination of all of these parameters are to make a good antibody, but I think slowly but surely we're getting there. Developability parameters so are kind of also interconnected, and they are they can be computationally determined on the sequence and structure level. So in this talk, we will only talk about computational developability profiling, although of course I know that essentially some of these parameters can be determined with more or less overlap with the computational domain. And they're also interconnected with one another. There has been an increasing interest in computational developability profiling with numerous papers on that topic, both reviews and otherwise. And now I'm going to briefly review the recent progress in computational developability profiling. So there are several studies out now that are advancing the field in computational developability profiling, but many of them look at the currently published therapeutic antibodies and define them kind of as the ground truth of what is good developability and what is not good developability. But what we lack is kind of, so these are about 100 antibodies or more, but what we lack is this underlying developability distribution, which you see here in the light green, which is actually the developability landscape. So what's the potential developability space in which we navigate? And we currently only have covered a very small space of that. And as I alluded before, there is a lack of harmony on which developability parameters matter. So there have been in silico tools for assessing developability, and there were, in 2017, people looked at 12 developability parameters or biophysical measures, then 5 in 2019, then 9 in in another paper, then 24, then 900, and then so it really it varies a lot across different publications, and there's no real consensus on the right number. And what we think is also missing in the field is the comparison of these more, let's say, non-native developability distributions to the native native repertoire, by which I mean just immune receptor sequences isolated that you can, for example, find in public databases. And why this is important? So we all, when you isolate antibodies from a mouse or a human, they usually have less developability issues, and it seems to be becoming more and more clear, and especially they seem to be less immunogenic than those that are isolated by various in vitro display profiling technologies. And it seems to be that the native space might not be as restricted as was previously seen. And yeah, I think I can move forward here. And I think it has, and there are even companies are built on the concept that we can isolate that we profile natural immune receptors to isolate good binders for a given disease. And it's also been shown that numerous therapeutic antibodies in the market are actually close to sequences that are just in public immune receptor databases. And also, for example, natural antibodies have been found to be that could be important, for example, for various disease treatments, such as Alzheimer, for example. So overall, I think there's a good amount of evidence that we should, before we look too much at the non-native space, just profile the natural repertoire space. Could be a good idea to just get an idea what's the capacity of natural immune receptor repertoires, so that we can then know where we have to go in and artificially tune antibodies to make them even better. And as we have also heard before, developability is a multifactorial measure. You can change one biophysical property, and it will then change others as well. And the main problem seems to be also that many of those developability of biophysical parameters actually lie in the binding region. And if you want to increase binding, then you also change developability. So it's really a multi-dimensional problem, as Pietro has also explained before. So there is this need for multi-parameter developability analysis. So the research question that we, knowing the background of what I just told you, our research question was, what is the developability landscape of the natural antibody repertoire, and how does it compare to what we call non-native developability spaces, such as those of humanized mice, patent-submitted antibodies, and therapeutic antibodies. And we're going to look at that in terms of correlation analysis, or what's called here redundancy, sensitivity, where we computationally mutate the antibodies and then look at how sensitive the different biophysical parameters are to mutation, and then also to what extent can each of those biophysical parameters be predicted. So what do we use? So we used very large. So we used OAS to and as well as our own data to assemble 2 million antibody sequences, about 200K per isotype. And then we have humanized mouse dataset, which is public. Then we use the patent submitted dataset, about 200,000 sequences from kindly received from natural antibody, and then also the Thera subtype database with 390 sequences. And we have both human. So in the native dataset, we have both human and mouse dataset. Although for the mouse, we are not as complete in terms of isotypes because of data scarcity in terms of different isotypes. And then we looked at 86. So again, developability parameters that are both on the sequence and structure level. So for all of those sequence numbers that you see, so 2 million, the 200,000, and so on, we calculated also their structure using A-body builder, and and then calculated also structure-derived developability parameters. So this was quite a significant computational effort to assemble this data set. So in the end, we have basically, let's say, it's just based on the native dataset, a matrix of 2 million rows, so 2 million sequences, then times 86 developability parameters, and that we have both, and that we have for sequence and structure and for the non-native datasets. So, and just some nomenclature, I will call, so for us, for a given antibody, a developability profile is just the vector of the 86 different developability values. So it's just one row of the explained. So the first question that we had is, how do sequence and structure-based developability parameters, now abbreviated DP, compare to one another? And for that, we saw that sequence-based developability parameters, which you can see on the left side, show higher correlation with one another than structure-based developability parameters. So there seems to be, let's say, less redundancy on the structure side compared to the sequence side, which one could understand because the structure-based ones are maybe a bit more complex and maybe based also on non-linear dependencies in structures. And we see on the right, if we correlate those 86 parameters with one another, you can see that there are some classes of high correlation in red, some of low correlation in blue, and some but many don't correlate much with one another in gray. So there's some developability parameters that we looked at seem to be more or less redundant, i.e. highly correlated, but many of them shown in gray correlation are also seem to be independent of one another. And this dependence and let's say or pseudo-orthogonality of sequences one can determine via Greiff's theoretic approach, where you can turn these heat maps that I just showed into a network and then isolate those parameters that are more or less correlated to other ones, and this allows us to isolate those ones that are more independent of one another, and these we call dominant nodes that are more independent of other parameters. And this can this approach is called, if you look in the left bottom of the slide, this is called minimum weighted dominating set. So this is a graph theoretic approach. So you turn your correlation network or your correlation heat map into a network, and then you can isolate those parameters that are more independent of the other ones. And this is then a subset of those 86. And we did this both by sequence and by structure. And you will see later why this matters. And we also saw that these vary, for example, by isotype, I think also. So there are some interesting trends to that. So, but then the next question was, can we distinguish actually antibodies by species and chain by the developability parameter profile? And again, the profile is this vector for each antibody. And what we could see is that that we see both chain type, and so VH and VL, but also species specific, so mouse and human developability signatures, both on the sequence and the structure level. So you see that there is a clustering of the and the mouse, more or less clearly, but yeah. And especially also a clustering of VH and VL. So both human and mouse seem to have different developability spaces, but also the different chains. And I'm going to get more into the space thing in the later part. So here we show again a principal component embedding of this developability matrix, and you see really that the heavy and the light chain developability space really clusters apart from one another with the light and the dark green. And if we then separate those spaces, we see also within the chains also human and mouse separate away from one another. And I guess that's also, I guess it's kind of a nice thing to see why we actually need humanization to be less immunogenic and so on, because developability spaces really seem to be different. And then an important question that we had based on our background in my research groups, that we mostly look at binding. We know that very similar sequences can exhibit very different binding and the other way around. So we wanted to see, is sequence similarity correlated with developability similarity? And what we see is that it's largely, so there is no high correlation between sequence similarity and developability similarity, which you can see in this plot, but maybe we focus on the structured parameters, which you see on the right side. And there we see on the right side in green sequence similarity, and the higher the green, the higher should be the sequence similarity. And you see that this does not correlate with clusters of high developability similarity, which you see on the left side, which should be black. So when you see black clusters, those black clusters do not correlate into dark green clusters. So and to some extent we see this also for the sequence-based developability parameters, which again I think has important implications for antibody engineering, that just changing one amino acid can already change the developability profile, which we also know from real world that this can happen. But it's nice to see this on a very large scale here. And we quantify this once more, that this correlation of, if you look on the plot labeled with C, that the correlation between developability space and antibody sequence similarity is very low, with a Pearson value of 0.2. So then we went to sensitivity, and this became a bit more difficult. So what we did is, so we wanted to kind of understand what happens what happens to a developability parameter if we change the sequence by one amino acid? Does the developability parameter change a lot, or does it basically stay constant? So this is shown here. And the important thing to understand is that we looked at two different sensitivity metrics, which are measured by kurtosis, which translates into sensitivity, or average sensitivity, and we also look at the whole range, so min to max, at the potential sensitivity of a developability value. And this we only did for sequence-based developability parameters, which I'm going to give the reason for that in a minute. So this would be an example case of how this average sensitivity and this range of the sensitivity would look like for different parameters that we looked at. And basically what we see is that the sensitivity varies by developability parameters, and it also varies within those developability parameters by antibody region. This variance is not consistent across the different developability. You can see the box plots do not follow the same trend across the different regions. And we wanted to do this also for the structure, but what we found is that if you change an antibody by one amino acid and then infer a structural model again, you do not see a difference based on the structural model. And we try to also, okay, is this only based on the structural models, or is this also, do we also see this from experimental structural data? So we went to structural databases and looked for mutants that are only one amino acid away, and also there it was very hard to see a difference in terms of angstrom between mutants of one amino acid, at least the ones that we looked at. So therefore we didn't perform developability analysis here, because it would have just been the same thing. That said, I think if in the future we had more experimental data of mutants, both experimentally, and if we had also higher sensitivity computational structural models, I think this would be very interesting to look at. I will tell more about the structure difficulties in a minute. So I will try to speed up a bit. So we also looked at predictability. So can we predict unknown developability parameters based on an antibody given its known developability parameters, was the question. So this is a complicated slide, but basically we had so here we focus on the first. So we had two machine learning tasks. We had one machine learning task where you see on the left, we just delete one column, so an entire developability parameter, and we just try to predict it based on the others. So we delete the one, which you see as an X, and then we try to predict back those X's. And the machine learning task 2, which you see on the bottom, is just randomly deleting developability parameters in this huge matrix that I mentioned before. So the task 2 we haven't finalized yet. So I'm focusing on task 1, where we used as encoding both developability profiles as well as also protein language models. And the one that we used is here, the ESM1B. And then we try to predict developability parameters based on these two embeddings. And the prediction was multiple linear regression. And yeah, so we used ESM1B as mentioned. Yes, I can move forward. Yes. So what we saw, and we did this by sequence and by structure based parameters, and we sampled antibodies in different numbers, so 5, 100, 500,000, 10,000, 20,000, and then we of course repeated this several times to get this variance. And what we see is that the protein language model is much better than just training on the developability parameters per se. So the protein language model picks up on some kind of sequence rules, I guess, which is very nice to see from the protein language model side, because since they're becoming so en vogue. But what we see on the structure side, we don't see much of a difference between those two. So the structure ones are very hard to predict if we want to predict one developability parameter based on others. And interestingly, for the if you look in the upper plot, we see that developability prediction of those orthogonal of this minimum weighting dominating set developability parameters, so those orthogonal ones, those less dependent ones, is harder than of the non-orthogonal ones. But this is only a problem for the non-protein language model embedding. If we embed with protein, if we use the protein language model encoding, then there is no difference between those two sets, which is also quite interesting, which kind of tells us that that how we think of orthogonality in the correlation space does not translate to the protein language model space, which is a much higher space. So I think theoretically that's quite a nice fun fact to think about. Anyway, I have to speed up. So just at the very end, we also asked, how do non-native antibodies relate to native antibody developability spaces? And basically, I just maybe go to this slide where we have in the background blue the native space, and then in the foreground we have the humanized mouse, the pattern submitted antibodies, and the therapeutic antibodies. And you see that all of them are in the natural space, almost not going out of the natural space. And this is for the heavy chain. And so this is for the developability embedding. For if we use the protein language model embedding, this holds true as well. And this holds also true for the light chain case, for both the language model embedding and the developability embedding. And then we also looked at, is there an overabundance of liability motifs in native antibody repertoires compared to non-native ones? And we also could not see any difference between a native and non-native. And then very briefly, because I think it's really important, because people will ask, why did you use Antibody Builder and not the new tools, and what's the dependence of your developability parameters on structure prediction tools? So first of all of our analyses were done with heavy and light chain, or most of the analysis were done with single chain, so heavy or light chain, but we saw a great correlation between single chain versus pair chain. And the one thing that we need to be careful about, that what we see here, and was also seen in a previous maths paper by Jane et al., is that the structural developability parameters depend on the structure prediction tool. We see very low correlation of developability, of structure developability parameters across tools. So, and by that we mean Antibody Builder 2, Antibody Builder, IgFold, both versus the crystal structure and among each other. And then we said, okay, is this based on the structure prediction tool, or is this just based on the fact that we are just looking at one point in time of the dynamic space of how proteins actually behave in solution? So, and what we could see actually that that actually models from an A-body builder structure or an experimental structure in the developability space, actually, then they do overlap, which we see. So, so we calculated across the molecular dynamic spectrum parameters of, I think, 5,000 different structures, and then compared them starting from an A-body builder structure, and then we can see that those developability spectra actually do overlap. So see a low correlation of structure tools among each other for structure developability parameters is most likely that we're just looking at one point in time and not at the MD spectrum. And with that, I would like to conclude that we found that sequence developability parameters seem to be more redundant than structure-based developability parameters. And we can predict them better. Protein language models capture sequence-based dependencies that enable prediction of even orthogonal developability parameters. And we see that non-native sequences lie within the native space, but they also show traces of human intervention. I didn't go into detail into that, but if you look back at when you see the recording, you can see that there is some kind of correlation in the non-native space, which we interpret as kind of a trace of human intervention. What we want to now look at is, can we predict a non-native developability parameter profiles from the native ones? And we need to improve a bit better our understanding of structure-based developability parameters in general, I think. And with that, I would like to thank especially the team that has worked on that in RED, as well as our funders. And thank you very much for your attention.
Great. Thank you very much, Victor. I think in the interest of time, we're going to shoot one question. Would it be better to define and refine the DP for each isotype rather than considering all of them? Aren't we just adding noise?
Can you say once more? I have to find the question.
Would it be better to define the DP for each isotype rather than considering all of them?
So what we saw is that that the DPs are largely consistent across isotypes. We also showed this with the protein language models, that there is not much of a difference across those isotypes. Yeah, that's what we see. But we didn't include we included it just as a label that different isotypes can increase or decrease half-life and so on. But for the parameters that we looked at, we didn't see a difference between those isotypes.
Cool. Thank you very much, Victor. Thank you for this talk. There are more questions, but I think that's like, if anyone is like,
I will answer.
Yeah, absolutely.